Okay, so we understand now somewhat that how transcriptional activation happens, right? Now we know about an enhancer and we know about transcription factors that bind to the enhancer, which recruit the mediator complex, which in turn recruits the RNA polymerase 2 in the promoter, and then transcription happens. Now, in very plain language, if you know of a signaling molecule or a transcription factor and you want to know which genes transcription it regulates, there is straightforward assay. Well, how do you do it? You know, if it is a signaling molecule, you simply add the signaling molecule to a cell culture system or a actual animal ex vivo organ culture system. There are many systems that simulate the in vivo actual environment, and then you quickly do transcriptomic analysis, either microarray or RNA-seq, and then you can understand that which genes are transcribed because of the action of the signaling molecule. Because the ligands are commercially available proteins, you can simply add it and you can do it. It is a little bit more complicated with transcription factors simply because when you add the ligand, the signal transduction process immediately starts. But that is not the case with the transcription factors, because you cannot add the transcription factor to a culture because they are nuclear protein. They do not act from outside of the cell. You have to express the transcription factor. So if you want to do a really good job of it, you will have to know accurately when the transcription factor that you are introducing is first expressed in the cell, and then you have to use that as your time point t0, and then you look at the transcripts at different points after that time. That is possible. With loss of function, it is even more difficult, because then you will be looking at which are the transcripts which are disappearing because the loss of the transcription factor. So like if you were talking about making a knockout mouse, the mouse is knocked out at the stem cell level, right? So whatever its effect is, it is starting from the stem cell level, and then at the zygote level, and then at the blastocyst level, and then perhaps you are analyzing the transcripts made in the lymph bud or in the neurons or something like that. Many days have passed in between the loss of transcription factor and when you were actually looking at it. Then therefore there will be the effect will be will not be very straightforward to analyze, because please understand that a transcription event typically takes half an hour to an hour to happen. So when you are looking at it like say the 30-40 days after the transcription factor is knocked out, then so many rounds of transcriptional regulation has happened in between, and these are all hierarchical processes, right? Like one transcription factor may influence the expression of 20 genes, okay, of which some will be transcription factors that will each of those can influence the transcription of another 20 genes each. So that way it gets more and more complicated with time. So but nonetheless there are some methods available. Now flip the question, that you know that a gene is only expressed, say, in limb mesenchymal cells or in eye, like PAX6. PAX6 is only expressed in the eye, right? It is not expressed anywhere else. So how would you know? If you know of a gene that is transcribed in a spatially and temporally regulated manner, and therefore that is a gene that you really want to study, how would you know which are the transcription factors that are responsible for its transcription in a temporally or spatially restricted manner? That is a question that we have not even we have begun to ask that question, but we are nowhere near understanding how it can be done. So to get you to this level of understanding, I'll just take you through some of the basic information. So first of all, if you want to know about transcriptional regulation and its implication in physiology, there are some beautiful review articles that you can read, and this one by Richard Young is a relatively recent review article, and it is pretty good. If you can read it and if you can understand it, that will be very good for you to understand. Now, let us approach the question that the question of gene regulation. One term that you will hear over and over again is the cis regulatory module. Okay, so when you are talking about activated transcription, there are two components. One is the enhancer, which is cis to the DNA that is being transcribed. So therefore, this is called the cis regulatory modules, and the trans factors, meaning the transcription factors. Okay? Trans meaning they are not part of the DNA; they are not intrinsic to the DNA. So there is like when it comes to defining the cis regulatory module, there is a constant back and forth between experimental approaches and computational approaches. So I will give you some basic ideas. Forget about how you got to know it, but say you have a hunch. Okay? You have a hunch that the enhancer will be in the first 1,000 bases upstream of the hunch. Just a guess. If you want to see if that is true or not, then what you can do is you can take that length of DNA right before the transcription start site, or including the transcription start site, like some 2,000 base pairs, and you clone it in a vector that already has a built-in reporter gene. Okay? Now, because you have the transcription start site already there, so you will be able to express the reporter. This plasmid should not have any promoter or enhancer element of its own. It should come from the DNA that you are investigating. So you take that piece of DNA, put it in this plasmid. In this plasmid, hold on, let me just get my pen. Yeah. So this is the plasmid where you have the reporter gene, and the reporter gene should not be preceded by any promoter enhancer. Now, of course, it will have its antibiotic resistance cassette and origin of replication and all those things for bacterial replication and selection. And in this thing, then you put this piece of DNA right upstream. So this will bring in the promoter site and possibly the enhancer site. Now, this you can do both in vivo or in vitro. In vivo is like in actual animals or in cell culture system. Now, this is the example given is like a gene that is can be stimulated with some chemical. Okay, I do not remember exactly which chemical it was, but some chemical. So you clone that and you make different lengths of this. See, like this part is the same in all the constructs. This part is varying, full length, a little bit less. And then you transfer it into the cell, it's a mammalian cell, and then you add the stimulus which this enhancer is supposed to respond to. Okay. And then you see where the reported gene is coming up. If you see that the reported gene is coming up when you have construct 1 or 2, so you know that and with 3 it is already gone, and 4, 5 it is completely gone. So you know that this part of the DNA is likely to be important for the enhancer activity. And you can do the same thing in animal system as well. I'll show you. I'll give you an example. But this is not as straightforward as you can think, because the transcriptional regulation is enormously complex. Look at this particular example, PAC6. Okay? PAC6 is expressed in the eye, but it is also expressed in the pancreas, it is expressed in the lens and cornea, it is expressed in the retina, it is expressed in a part of brain called telencephalon, and rhombomeres, and in many other places. And what is interesting is that for different places of expression, for pancreas, there is a separate enhancer. For lens and retina, there is a separate enhancer. For telencephalon, there is a separate enhancer. For retina, there are these two elements. And for other parts of the brain, there are other elements. And these enhancers are such that the transcript itself changes. So there are three kinds of PAC6 transcript. If it is the lens or the pancreas, then it starts with this exon 0, the longest mRNA. If it is the transcript for telencephalon, then it starts from exon 1, and you have this mRNA. And if it is for retina and other things, then it starts from this alpha exon, and then it continues. And as you know, that sometimes enhancers can be within the gene as well. So in this case, this is an example of that. That it must be influencing transcription to start from here. Okay. So therefore, now you see that how complicated it is. That there are some there are more than one promoter-like sequence. There must be a promoter-like sequence here, there must be a promoter-like sequence here, there must be a promoter-like sequence here. There must be a promoter-like sequence here. So now which promoter the POL2 will be recruited to, even that can be dictated by the enhancer. And each of these enhancers, the fact that they are different enhancers, tells you that they all bind to their own favorite transcription factors. That's the reason why when the gene is being expressed in the pancreas or in the lens, uh, there are certain factors that are present only there, and the transcription factors, because see, the DNA sequence is the same in every cell, right? So there are certain factors, let us say A, B, and C. If they are present only in the pancreas, then the pancreas, then PAX6 will be expressed in pancreas. And if D, E, and F, they are present, say, only in the lens and cornea, and not in any of these other tissues, pancreas, retina, etc., then PAX6, the retina, lens, and cornea version will be produced only in lens and cornea, and so on and so forth. So therefore, defining the enhancer is not as simple as one may think. There was a very powerful kind of technology that was used in the 90s called enhancer trap. Enhancer trap is a very interesting experimental concept. That is, you will take a reporter and it will put some DNA sequences that can allow it to be integrated in the genome. Okay. Somewhat randomly. Okay. Now, this reporter sequence, if it by chance gets recruited close to a promoter enhancer, then you will get a reporter expression. Okay. Now, there is no guarantee, but if you get to see reporter expression and it is in your favorite tissue or cell type, then you can see where it is being, where it has been integrated. It is very simple to determine where it has been integrated because you know the sequence of the cassette that you have inserted, like that you have prepared, right, for insertion. So because you know the sequence, so you can sequence from here and you can sequence from here, and then it will go into the genome where it has been inserted, right? So because then you will get to know the Franklin sequence, so you will have a good idea that where the integration has happened, and you can take the region around it and try to figure out which part of the genome has this enhancer activity. It is easier said than done though, because each of these will require that you have to create a transgenic mouse. Remember transgenic we have talked about, where you inject in the female co-nucleus. And you're talking about few months of experimentation before you get to see if the reported gene is at all expressed in your tissue of interest. So it is a process that has been used, and some enhancers have been discovered using this, but it is not a very straightforward. But if there is a reason for you to suspect that a particular piece of DNA is an enhancer, and say, let's take the example of TAC6 in lens. Okay. So you want to know that, say you know the enhancer sequence. Right? So you want to know what are the transcription factors that are regulating expression of TAC6 in lens. So what you can do is you can take nuclear extract from lens, and you can take the TAC6 enhancer region, which you have already known. Okay, you know this sequence, right? It is there on the map, meaning you know the sequence, and you make that DNA radioactive. Okay, so the radioactive piece of DNA will have its migration based on its size on on an agarose gel. Now, in the other lens, what you can do is the other lens within these. Okay, these are called lanes. Okay, and we are talking about the lens tissue. Okay, sorry for the confusion. So you take nuclear extract from lens tissue and you incubate it with the radioactive piece of DNA, and you run it on a gel. Okay. And when you run it on a gel, if you see if there is any protein in that nuclear extract that can bind to your piece of DNA, then if you run it on a gel, then the DNA will migrate slower than it normally was migrating, because now its size has increased. So you will see that it is migrating slowly, and that's called electrophoretic mobility shift assay, because the normal mobility is this, it has shifted. So you know that there is something in this protein solution that can bind to the enhancer. Okay, and you can do fractionation scheme, you can actually purify it, and then you can identify the protein. So that is something that has been done for identification of transcription factors when you know the enhancer. Okay? Or you have a reason to suspect that this is an enhancer. You can test it kind of in vivo, like in a cell line. So you have I do not know if it actually exists, but let us say that you have a corneal cell culture. Okay. So there, what you can do is you can take two different plasmids. One plasmid will be expressing, it's an expression plasmid for the protein X, with which you got this electrophoretic mobility shift. Okay, so now you want to see whether it can really act as an enhancer, as a transcription factor for a particular enhancer. So you will take one plasmid where this X will be expressed, and you will take another plasmid with the reported gene, and then you have the X binding site. Okay? So this is your enhancer area, and then the promoter will be somewhere over here. Now, if you take two such experiments, where in one you transfect this plasmid 1 plus plasmid 2, which carries X, and in another one, plasmid 1 plus plasmid 2, but it does not carry X. Okay? Everything else is same, just this site is not there. Okay. No, you should not do it that way. You should not do it that way. I take it back. I'm changing it. Plasmid 1 that expresses X plus plasmid 2, or plasmid 1 that does not express X. plus 150. If in here you get a lot of reporter expression and here you don't, then you know that X indeed does something to this X binding site to increase the expression of your reporter gene. So you have now identified a combo of an enhancer element and its corresponding transcription factor. So first you have to have a reason to believe that a particular region of the DNA is the enhancer. Then you can do EMSA test to identify EMSA assay to identify a protein or proteins that can bind to that DNA region, which is the putative enhancer. Putative meaning likely enhancer. And then you can do this kind of a reporter assay to cement your conclusion. It is still not exactly in vivo. It is in cell culture system. There might be variations, but it's a good first experiment. Now, so that is what you can do with the cis regulatory module. And when it comes to the trans factors, the transcription factors have an advantage that they have this domain-like structure, the DNA binding domain and the activation domain are structurally and functionally independent. So often what we will see in certain labs, certainly in my lab, what we do is we have identified a transcription factor. We want to ensure that this transcription factor really drives expression of 5 genes. Okay. I have, for some reason, I know the 5 genes that I should look at. One is to simply misexpress the transcription factor in the particular tissue, but sometimes when you have the normal dosage of transcription factor already present, which is particularly the case with chicken, then if you misexpress it, it may or may not give you dramatic result. But if you can somehow increase the efficiency, which is, you make a constitutively active form, then what you can do is you take your favorite transcription factor and you use its DNA binding domain and you fuse it with the GAL4 activation domain. GAL4 has a very strong activation domain. So if this transcription factor is likely to increase the expression of 5 genes, then this chimeric transcription factor, you are very much likely to see very high level of expression of your transcription factor. If you want to read more about it, like how this modularity of transcription factor is used in biotechnology, you can read these links to learn about it on TETOP system. They're exactly this kind of things are done. If you read it and you have any question, please come back to me. I will explain it, but it is not for the general purpose. Now, say you know the transfactor, okay? You know the enhancer. You now want to know what kind of sequence the transfactor binds to. So please try to understand that we are slowly narrowing down the scope of the analysis. First, we wanted to know if there is an enhancer, like if I can identify an enhancer. There are many methods, like most of the time you chance upon an enhancer, or you simply take a guess that it will be the first 5,000 bases before the transcription first starts or something like that. There is no hard and fast rule how you like based on which criteria you will suspect something to be an enhancer. That is a topic that is under active research, and I will talk to you about it in a little while. But say you have an enhancer, then you want to know which transcription factors bind to this enhancer, so you can do EMSA test. Then once you got the transcription factor, then you want to show that it actually acts as the transcription factor for this particular piece of enhancer DNA. So you can do the transfection assay like as we saw here, and you will get it. Okay. If you want to get more emphatic result, then you can swap and make these hybrid transcription factors. You can do. Now you want to know that within the enhancer, where is it that that specific transcription factor is binding to? This is very similar to your restriction enzymes, right? Restriction enzymes recognize a 6-base sequence or 8-base sequence or 4-base sequence. Transcription factors typically will recognize a sequence of 10, 12, 15, 20 base pairs, and this is not more than that. So what we will do is what is referred to as DNase 1 footprinting assay. I'm pretty sure that you have been taught about it, but I will still talk about it another time. This experiment is not as straightforward as you think. Look at the radioactivity. The radioactivity is only on the 5' end on one strand. Okay? And then you have two systems. In one, you have your protein of interest added, and in the other, you do not have your protein of interest added. Okay? Then you subject it to very low concentration of DNase 1. If you put a lot of DNase 1, everything will be digested. Very low concentration of DNase 1. So that only by chance some of the sequences will be stopped. Okay? It is purely a chance thing. Use therefore very low concentration. Now, because you are using such low concentration, where the protein is actually bound, that specific region will be occluded, will sterically block the DNase 1 to go and cut that piece of DNA. Okay? So therefore, if you are cutting randomly, you will cut, in all these different kinds of positions, so you will get a ladder of DNA. But because of this occlusion, so this and this cut will not happen. Like this and this cut will not happen here. Okay? The rest will happen. So you will see a gap in the bands of radioactive bands, and this is possible only because you have labeled only on one end of the double-stranded DNA. Now, in the same gel, if you run a DNA sequencing in parallel, where it will start from here, then you will get to, that will start from here. Then the sequencing gel is also a reflection of the length of the DNA, right? So if you map exactly here, and you can tell exactly which sequence is being bound by the protein. So you will not only know how far it is from the 5' end, you will also know that exactly what sequence it is where the protein is binding. So this is called DNase 1 footprinting assay. So in the 90s, this kind of assay has been done for many factors. Okay? And for the same transcription factor, it has been done in multiple locations within the genome, multiple different organisms for the same gene, like same gene, multiple different organisms, or same organism, multiple different genes. Okay? And then you get to see that where is it that this particular transcription factor binds to. And just like the way we have done it for Tata box, you get to see the frequency of bases from 5' end to 3' end, where the transcription factor is binding to, and you draw the letter based on, like the size of the letter will be based on the frequency. If it is, like 80% like this, if it is a C, it is 100%, then it is the biggest. So if it is, C in position number 3, in 60% cases it is C, 40% cases it is T, then you write it like that. This is called what is referred to as position weighted matrix, or PWM. So such position weighted matrix for many transcription factors have been determined, and that is logged in a database called transcription factor database, transcription factor binding database. For many transcription factors it is there. But if you look in the literature very carefully, you will see that data from transfact database has actually not been used much in experimental purposes. Like looking at transfact database, someone predicted the binding site and then actually tried to see if either the transcription factor actually has a role in regulation of the gene of interest, or it actually binds to exactly that sequence in vivo. Those kind of experiments, at least I could not find in the literature. Therefore, I am assuming that they have not worked in most of the cases. Okay, and if it is the case, I am not sure that if that is the case. Maybe no one ever wanted to use the transfer database, so there is no literature. But that is unlikely. if it was used and no, informative, no information could be gathered from it, then it will not be reported in the literature. And if that is the case, the one likely reason why it may not work is that this entire data is generated using naked DNA, which is not the case in vivo, right? In vivo, DNA is in the context of nucleosome, so the sequence, the specificity that you are assessing based on PWM may not actually be valid when you are trying to look in vivo. So that is a problem. So to circumvent that problem, now we have another method, which is called the ChIP-seq method. Now, ChIP-seq method is very good if you are doing it this way. That if you want to uncover the binding site for a particular transcription, you know the transcription, then what you can do is that you will have an antibody against this transcription factor. Sometimes you can also use a fusion protein against whom there is a good antibody present. But you have to first demonstrate that the fusion transcription factor actually works just like the normal transcription factor. If that is there, then you can use the antibody against the fusion part of the transcription. So either it doesn't matter. So what you can do is that you isolate the nucleus and then dissolve it and add your transcription factor, or add an antibody against your transcription factor, cut the DNA very gently, either using sonication or using DNA or something like that, and then you immunoprecipitate the transcription factor. It's very simple. Antibody will bind to the transcription factor, and the antibody can be further bound with a secondary antibody, or the antibody itself can be tagged with something called saccharose or agarose, which is insoluble. So you will simply spin it out. The agarose will come down, but that is covalently linked with your this antibody, which is in turn very tightly linked with the transcription factor, and which in turn is bound to the DNA. And nowadays, particularly with advent of next generation DNA sequencing, you can very easily get a very large number of such fragments that came down with the transcription factor, and you will be able to know that exactly where the transcription factor binds in the in vivo. But there is one very interesting thing that you would want to remember, that a typical footprint of a transcription factor will not be more than 15 to 20 nucleotides. But the ChIP-seq data output is typically 200 nucleotides. Therefore, when you get the ChIP-seq output, you get sequence information more than exactly where the transcription factor is binding to. So please remember that. And from that experiment, you will get to know a whole lot of things. That where is the transcription factor bound? What is its DNA binding affinity? There is just a quantitative measure of the DNA that came down with it. The more the affinity is, more DNA will come down with it, and there can be some normalization. What genes might it regulate? Which is not really an exact science, because as we know that enhancers can be inside the gene, outside the, like upstream of the gene, downstream of the gene, sometimes very far away from the gene. So just because you have the information that where this particular protein is binding to, you may not necessarily understand that which gene it regulates. But in most of the cases, it will regulate a nearby neighboring gene. So therefore, you can guess, but it will still be a guess. You have to experimentally prove it. Okay? And what are its partners. So once you know one transcription factor, then there are many ways of determining proteins that physically interact with it. Or remember, I told you that you will get 15 to 20 nucleotides, the actual 15 to 15 to 20 nucleotides, but you will get information of 200 nucleotides. if you know the binding site for other transcription factors, you can simply find it there, and then you can know what are the partners along with which this transcription factor binds to its cognate enhancer. And nowadays, this is being facilitated by very large-scale experiments, which are all being archived in UCSC genome browser. UCSC, University of California, Santa Cruz, they have a a server where all the data is input. So ChIP-seq data for many of the transcription factors have been done, and that are mapped onto the genome. So because the human genome sequence is known, so you can map the ChIP-seq data onto that. Okay? And once you do that, you can get this kind of transcription factor binding sites on there. But if you just go and play with the UCSC genome browser, ask to show the ChIP-seq data, you will actually get to see it. Okay? So here. And these ones which will map next to a DNase cluster, the DNase hypersensitivity cluster. When it is DNase hypersensitivity cluster, it is a measure of open chromatin structure. So if the transcription factor binding site coincides with an open chromatin structure, chances that it is an enhancer is even more. Nowadays, there is another new technology called ataxic. Okay? This is a measure of just a simple question, where the DNA chromatin is open, where the chromatin is closed. If you find a correlation between open chromatin region and ChIP-seq data, then you are more or less certain that it is an enhancer. You cannot immediately predict that it is an enhancer for which gene, so this DNA binding site, etc. This may be an enhancer for superoxide dismutase, SOD1, but it may not be. Just because it is close to it does not mean that it actually is regulating. All you know that it is regulating something 2 megabase downstream. Okay? So that's what it is. So let us stop right there. This is a little bit complicated thing. So think about it, ask questions during discussion, and then we will move to the next part. Okay. Thank you very much.